Okay, so hopefully this goes smoothly, but for the first problem it asks us to perform a cross for 5 different alleles and assume that it undergoes independent assortment. So for 1A, it asks us to find heterozygous A, homozygous B dominant, homozygous C recessive, homozygous D dominant, and homozygous E recessive. Oh, and I guess before I say that, I should say that I made 5 different Punnett squares, each for the different alleles, if you can see that, and then I labeled them on this side: homozygous, heterozygous, and homozygous recessive. So for A, I found out that specific genotype would be a 1 in 128 chance. For B, it asks us for homozygous A recessive, homozygous B recessive, homozygous C recessive, homozygous D dominant, and homozygous E recessive. And I found that genotype to have a 1 in 256 chance. For C, it asks us to find homozygous A dominant, heterozygous B, homozygous C recessive, homozygous D recessive, and heterozygous E. For that one, I found to be not possible because when I developed the Punnett square for the D allele, there is no possible way for it to have a homozygous D recessive. So for that specific genotype was not possible with their 2 parents. For 1D, it asks us to find heterozygous A, heterozygous B, heterozygous C, heterozygous D, and heterozygous E, and I found that to be a 1 in 32 chance. For number 2, I'll just read off of the paper. It says fill in the blanks. A, paired homologous chromosomes are sometimes called bivalents or tetrads. And I really hope that's right, because I don't know what else it would be called. Skipping to B, the structure that connects the centromere to the microtubules of the meiotic spindle is called, I don't know how to pronounce this, but I believe it's kinetochore or kinetoptor. Something I hope we're able to understand me despite my inability to read. Alright, 2C. A diploid fruit fly has 4 pairs of chromosomes. A fruit fly in prophase 1 has, and I found it to be 16 chromatids, while a fruit fly in metaphase 2 has 8 chromatids because it divided in meiosis 1. For 2D, null allele is a type of genetic cross that can, oh, I'm sorry, I skipped one. D, oh, okay. A monohybrid cross is a type of genetic cross that can determine if a purple flowered pea plant has the homozygous P dominant or the heterozygous P genotype. Cool. Then for E is the one that I started reading earlier. A non-functional allele is sometimes called a null allele, while a partially functional allele is called a hypomorphic allele. For 2F, it says Mendel's law of segregation involves 2 traits. Oh, I'm sorry, that's independent assortment. It should be independent assortment that involves 2 traits, while Mendel's law of segregation involves a single trait. And again, I may be wrong on that, but that's what I came up to be. Okay, for 3. 3 was the one that Sister Mills gave a video online. So I'll be real quick with this one. I mean, she did the same problem, just switched up the letters. Okay, so it says 3A, diagram all possible homologous chromosome arrangements in this organism during metaphase 1. Be sure to indicate which alleles are found on each pair of homologous chromosomes. Gosh, I hope it's. I'll just read them out, and then I'll show what I got. So I found 4 different arrangements for that. So I found, actually no, I'll just show you, because I mean she did all the work already, and so I'd hate to waste time in my video to go home and show that. So it's just making sure it's different arrangements. Simple as that. Okay, B, indicate all the possible gamete genotypes that can be produced in this organism at the conclusion of meiosis II. So there are 4 different gamete possibilities at the end of meiosis II, and that is that would be the homozygous dominant TRY, then got a dominant T allele, recessive R allele, and dominant Y allele. The third combination would be the dominant T allele and the recessive R and Y alleles. And then the last combination I found was the dominant T allele, the dominant R allele, and the recessive Y allele. For question C, I found there to be 12 sister chromatids during the time of metaphase 1. For D, it says how many chromosomes are found per cell in this organism during metaphase 2, and I found that to be 3 chromosomes per cell. Alright, so question 4, it told us that a horse has 32 pairs of chromosomes. The first part of question 4 asks us how many chromosomes are present per horse cell at the conclusion of meiosis I, and that should be 64 chromosomes because there would be 32 pairs of chromosomes, which we would just double that so we could see that we have 64 chromosomes, 32 from each parent. B, it asks us how many tetrads are present per horse cell during prophase 1 of meiosis. If we recall what tetrads are, let me get a definition. Anyway, I don't have a definition with me actually, but there should be 32 tetrads. I can't explain what a tetrad is at this moment, but because we had 64 chromosomes, we should have 32 tetrads at that phase in meiosis I. Okay. C, how many sister chromatids are found per cell in this organism during metaphase 1? And that is 32 chromosomes. Did I say that right? Yes. Sister chromatids per cell. Right. Per cell is 32 chromosomes because we already... We haven't split already. I might be wrong on that one. But I found 32 chromosomes, or... Oh, my bad. I skipped one. I was going to say, okay, we should have 128 sister chromatids in metaphase 1 because we have not yet split. And since we have 64 chromosomes, we should have 128 sister chromatids since we've already doubled but not yet split. Okay. D. Why do I have an E? Oh, I was looking at the wrong problem. But yes, we should have 128 sister chromatids during metaphase 2 of meiosis. In a horse cell. You know what? I did that for metaphase 1. I do believe we should only have 64 sister chromatids during metaphase 2 because we already underwent one split. D, how many sister chromatids are, nope, E. Okay, how many bivalents are present per horse cell during anaphase 2 of meiosis? And I do believe that is 0, actually. Yeah, I think that's 0. I'm struggling to find where my definitions are, but that is what I got. Okay, question 5 is our last problem. Okay, so I did the same thing. I made Punnett squares for each of the 4 alleles. Gosh, this is hard working with that camera. Everything's backwards. But I made Punnett squares for each of the different alleles and their combinations, and then I was able to just find my answers based off of that. So question 5 asks us what proportion of the offspring will be tall, round, green, and terminal. I mean, they're looking for a phenotype there, so they're really asking us, so tall and round are both dominant traits, which would either be homozygous dominant or heterozygous, would show up there. And then green and terminal are both recessive traits, so homozygous recessive. So we have a 3 out of 4 chance that it's going to be tall, a 3 out of 4 chance that it's going to be round, a 1/2 chance that it'll be green, and a 1/2 chance that it's terminal. If we just take the product of all that, we should have a 9 out of 64 chance that it'll meet the criteria for tall, round, green, and terminal. For B, it's looking for a genotype, which I found to be a 1 out of 32nd chance, or 1 out of 32nd chance, because it's a 1/4 chance that'll be a recessive T, 1/2 chance that'll be a heterozygous R, 1/2 chance it'll be a heterozygous Y, and a 1/2 chance it'll be a heterozygous A. C, it asks us to find the proportion for an offspring that has the heterozygous trait for all of them, which for all of them it's a 1/2 chance. So we just take product of that, and it's 1 out of 16 chance they'll have a heterozygous. It'll be heterozygous for every trait. D, it's again asking for a phenotype. What proportion of the offspring will be dwarf, wrinkled, yellow, and axle? If you take the probabilities for each of those traits, you find it to be a 1 out of 64 chance that it'll meet the criteria for all of those traits. Well, that wasn't the most smooth thing in the world, but that's it. Thank you guys.